Okay, so we're going to pick up here with part 5. This will probably be our last part. And we're going to talk a little bit about synapses, and then we'll finish by talking about um, summation, and we'll talk about different types of summation and things called EPSPs and IPSPs. So, our synapses, a synapse is generally just the space or kind of the gap between different neurons. And so you have a general synapse, you have a synaptic cleft, which is the actual physical space in between it, and you can name cells where they're located relative to the synapse. So are they before the synapse or are they after the synapse? If they're before the synapse, you call them a presynaptic cell. If they are after the synapse, you call them a postsynaptic cell. This will become more critical, talking about presynaptic and postsynaptic cells when we get to the autonomic nervous system and talk about that. Um, the autonomic nervous system, between sympathetic and parasympathetic, the length of presynaptic and postsynaptic neurons differs whether you're talking about sympathetic or parasympathetic. Sometimes they're also called ganglionic, preganglionic, or postganglionic. Ganglion meaning a grouping of cell bodies outside the nervous system. So sometimes you'll see that as well, but it'll become more important there when we get to the autonomic nervous system. Now, there's two types of synapses generally in terms of their structure and how they're working. We have electrical and chemical. Now, in electrical, we are using gap junctions to transfer different ions between cells. We know that gap junctions, they're a type of cellular junction. They're made up of a connexon, and then these connexons are made of individual connexins, and they allow for communication between adjacent cells. So you see these in cardiac myocytes within the intercalated discs of those cardiac myocytes. You have gap junctions, and this allows ions to spread very quickly from one cell to the next. And in the heart, you can imagine how this would be beneficial and necessary. It allows the heart to contract as a single unit, and sometimes that's referred to as a functional syncytium. It's able to contract as this single unit and not individual cells at individual times. That would make the heart's contracting very, very, it wouldn't be very smooth. Okay. Now, you can contrast electrical with chemical synapses, and chemical synapses are a large majority of our synapses within our bodies. These are utilizing a very specific type of chemical. These are neurotransmitters. Okay. Now, neurotransmitters can be things like acetylcholine. We have GABA or gamma aminobutyric acid. Our catecholamines can sometimes function as neurotransmitters, so epinephrine, norepinephrine, and we'll talk about these in just a little bit. And then you can take these chemical synapses and divide them even further. So you have ones called axodendritic and axosomatic. Axodendritic refers to the fact that the axon of one neuron is forming a synapse with the dendrites of another. Okay, and we'll take a look at those. And then axosomatic, you can maybe imagine based on how we define axodendritic, the axon of one neuron is synapsing with the cell body or the soma of another. So I believe we have a picture here. Here you have a axodendritic. So we have a axon here coming in, forming a synapse at the dendrites of this neuron. And we can also name these neurons relative, where they're located relative to the axon. So here's our presynaptic neuron. Here's our postsynaptic neuron. We know that this is postsynaptic because these dendrites are what are going to be receiving the action potential from one cell and bring it into this cell right here. So you know that this has to be presynaptic. Action potential is traveling along here. It will synapse on these dendrites, relay the action potential to the dendrite cell body, axon hillock, and then this postsynaptic cell will perhaps then become a presynaptic cell because it may have a synapse down here with another cell. So then here it would be presynaptic, and then there would be another cell here that would be postsynaptic. Okay? So here you can see the difference between electrical and chemical synapses. Chemical on the left, we have the action potential stimulating the release of neurotransmitters, and specifically you have calcium enter, cause the fusion of a synaptic vesicle with the axolemma here of the of the presynaptic neuron, releases those neurotransmitters into the synaptic cleft, and then can bind to our gates here, or our receptors. Now, this one specifically, these are called ionotropic or ligand-gated receptors, meaning that when they bind the neurotransmitter, they bind it, and then they themselves serve as the channel. So they're a receptor, and then they open and serve as a channel to perhaps allow sodium to enter the cell. Okay, and then here on the right we can see an electrical synapse. And again, we said that electrical synapses are formed from gap junctions. Gap junctions are connexons made of things called connexins, and allow communication between adjacent cells. So you can see that things would be able to flow freely between these two cells, and it is due to these gap junctions, these connexons, that is able to occur. And again, we said you find these in places like the intercalated discs of the heart tissue, so between adjacent cardiac myocytes. Now, our chemical synapses, so if we go back, our chemical synapses right here dealing with our neurotransmitters, we can sometimes receive multiple stimuli at a cell. So a postsynaptic cell may receive multiple stimuli from different cells. And we talked about earlier, some may be depolarizing, some may be hyperpolarizing. If one is depolarizing, we're going to call that excitatory. If it is hyperpolarizing, we're going to call that inhibitory. And you can take these excitatory depolarizing stimuli and these inhibitory hyperpolarizing stimuli, and you can combine them, or you can summate them together to maybe or maybe not produce an action potential. Now, we don't always reach an action potential, but we can sometimes if we reach threshold, right? And remember, threshold is about negative 55 millivolts or so. And so what you have are these excitatory and inhibitory impulses come together and may or may not reach threshold. So at the synapse, we have a couple of things happening. We have the action potential arriving that we talked about earlier. We have the action potential arriving at the synapse stimulates the release of calcium, or rather the entry of calcium, the opening of voltage-gated calcium channels to allow calcium to enter the synaptic bulb, or the synaptic knob, or synaptic end terminal. This calcium causes the synaptic vesicles to fuse with the plasma membrane of the neuron and to exocytose their neurotransmitters, which is usually acetylcholine, into the synaptic cleft. When these neurotransmitters enter the synaptic cleft, they travel across, find the postsynaptic cell, and bind to their receptor. And again, we said this is called a ligand-gated receptor because the ligand is the acetylcholine, the neurotransmitter, and then it opens up as a gate to allow perhaps sodium to enter the postsynaptic cell. Now, this synapse, this impulse, action potential, can either be depolarizing or hyperpolarizing. The stimulus can be. So if it is depolarizing, it is going to be called an excitatory postsynaptic potential. If it is hyperpolarizing, it is going to become an inhibitory postsynaptic potential, and you'll usually see them referred to as EPSPs and IPSPs. So if it is an EPSP, net depolarization, we open sodium and potassium gates to allow this depolarization to occur. If it is inhibitory, or a hyperpolarizing stimulus, it's going to open potassium channels, and we know that when potassium channels open, it leaves. It goes down its concentration gradient, and when a positive ion leaves the cell, it's going to make it more negative, and so that's going to contribute to it becoming more negative or hyperpolarizing. Okay, so in this instance, it would be inhibitory. It would be harder for an action potential to be generated because we're now more negative than we initially were. Okay, and these EPSPs and IPSPs, like I've said a few times now, they can summate, but the only place that it matters is at the axon hillock. So you could have EPSPs and IPSPs summate and be at threshold before the axon hillock, and you may not necessarily get an action potential because you have to be at threshold at the axon hillock. If you're not at threshold at the axon hillock, it doesn't matter what you were prior to that. You won't get an action potential. So it's very, very important to keep that in mind and understand that you have to be at that negative 55 millivolt threshold at the hillock if you wish for an action potential to be generated. Okay, so what you see here, here's an EPSP. We become a little less negative, more positive, coming up toward the threshold, and this is a depolarizing stimulus, so we're depolarizing then repolarizing. And if we have an IPSP, here we're hyperpolarizing. We're becoming more negative or less positive. We're moving further away from our negative 55 millivolt threshold. And again, this is because potassium channels open, potassium rushes out down its concentration gradient, taking with it its positive charge and making our membrane potential more negative. So in this instance, it would be more difficult for an action potential to be generated. But what you can do is take these IPSPs and EPSPs and combine them together. So if you took these two, these look like they're about equal in amplitude in either direction, either excitatory or inhibitory. So you could imagine if you combine this EPSP on top and this IPSP on bottom, you would get no net change. You would stay at about negative 70 millivolts or so. So the way that you summate can differ based on location and the type of stimulus arriving. So in temporal summation, what you're going to have many stimuli, whether they're inhibitory or excitatory, arriving at a single point on the postsynaptic neuron. Okay, so you can imagine one stimuli comes, and then another one comes, and another one, and another one, and another one, and so on and so forth. And these are going to summate and maybe reach threshold to produce an action potential at the hillock, or maybe not. Okay, so sometimes you can think of this as rapid fire, one stimulus, and then another one, all at the same point on the postsynaptic neuron. Now, with spatial summation, it is the kind of the opposite. Instead of one stimulus after another at one single point on a postsynaptic neuron, it's going to be spread out. So spatial, it's spaced out. Many stimuli still arriving, but at different areas on the postsynaptic neuron. Okay, but same thing. They can still summate, and we're only going to get that action potential again if we reach threshold. Where? At the axon hillock. It doesn't matter if you're at threshold before that or prior to that. It only matters if you're at threshold at the hillock. So what you see here, you can recognize this as being spatial summation. You have one stimulus here, you have another one here at different areas on the cell body, and at this hillock here, it's only going to matter if we're at threshold. Here we have temporal summation, one single presynaptic neuron transmitting many stimuli to a single postsynaptic cell at one single point. Okay, so temporal, just at a single location. Spatial summation, we are spaced out many stimuli across different areas of the postsynaptic neuron. And then here you can see a graphical representation of us summating our EPSPs and IPSPs. So maybe right down here we're at resting membrane potential, so we're at about negative 70. We had an EPSP, which is excitatory. We had another EPSP, and here we had an IPSP, so an inhibitory. Here we stayed the same. Here we had an EPSP. Here we had another IPSP, and then here we had a really big EPSP, and that got us to threshold. And when we were at threshold, we were at the axon hillock, so you get your whole action potential. Okay. So the thing to note is your EPSPs and IPSPs can be of different amplitude. They can be different in size. Your action potential is always going to be the same. Okay, it's not going to change. It's always negative 55 threshold, negative 70 or so resting membrane potential, and a max of maybe positive 30 or so millivolts, and then you drop down with your hyperpolarization. Okay, so here you can see how we're summating, how we're adding these different ones together, and it only matters where we're at when we get to the hillock. If we're not at threshold at the hillock, it doesn't matter if we were way back here. We're not going to have an action potential. Okay. So again, when you receive stimuli along these dendrites, travels through the cell body, you may be at action potential. You may be at threshold for an action potential way back here, but by the time you've reached the hillock, you've dissipated some of that, and now you're at, say, negative 40 millivolts. Well, negative 40 millivolts is 15 millivolts shy of our negative 55. Okay, so again, you wouldn't be at threshold, you would not get an action potential, and so you would have nothing be propagated along your axon of your neuron. Okay, so the last thing we'll talk about is neurotransmitters, and we'll talk briefly about the development of the nervous system, just very slightly. Acetylcholine is the primary neurotransmitter at our neuromuscular junction, the MNJ. It can have different effects depending on where it's located because of different receptors. So, in terms of its effect on cardiac muscle, whenever you release acetylcholine on cardiac muscle and it binds to receptors there, it's actually inhibitory. So it's going to be it would result in a parasympathetic response of the heart. So the heart rate would slow down, blood pressure would decrease, things like that. And the way that you deal with acetylcholine and you get rid of it is by the enzyme acetylcholinesterase. It breaks it down into an acetic acid and a choline molecule, and by doing so, you render the acetylcholine no longer effective. Okay, so you see this with muscles. So at the neuromuscular junction, like I said, it's going to be released from its synaptic vesicle across the synaptic cleft, bind to its ligand-gated receptor, cause that receptor to open, and you get a influx of perhaps sodium or something like that. And then when it's ready to be degraded, acetylcholinesterase will come by and do that. We have things like epinephrine, norepinephrine, dopamine, serotonin, histamine. Lots of these are derived from amino acids, so they're amino acid based. And amino acid based versus steroid based hormones and neurotransmitters will exert their actions differently. And you see a lot of these between neurons of the central nervous system. You can also have epinephrine at the heart muscle, like we talked about with acetylcholine. Epinephrine at cardiac muscle tissue in this instance would be excitatory. So you would have an increase in heart rate, increase in blood pressure, things like that. You have something called gamma-aminobutyric acid, or GABA. GABA is a very potent inhibitory neurotransmitter, so it dampens the nervous system. So many nervous system medications and drugs that are used to treat things like seizures, epilepsy, panic and anxiety disorders, things where we become too stimulated. Many of these medications that treat these things potentiate gamma-aminobutyric acid. So in some way or another, they either stimulate the release of GABA or potentiate it or mimic its effects. And by doing so, you get an overall inhibitory effect. So this is good for things like seizures, epilepsy, panic attacks, anxiety disorders, where you need to take the individual down in their stimulation. Okay. Then we have things like endorphins. Endorphins means endogenous morphine, so these are natural painkillers within our body, and these can be communicated and conveyed between cells as well. So the very last thing here we'll talk about is our development. Very briefly, we won't spend a lot of time on it. Our nervous system is derived from one of our embryonic layers called the ectoderm, and so the ectoderm also gives rise to the skin, but it also gives rise to our nervous system. You have things called neural crest cells, which can migrate out and become different types of cells. We have something known as the neural tube. This neural tube is a region in us as we're developing in embryos that will kind of form this. It will kind of arch backwards and form this tube. At first, it's called a neural groove, and then it becomes the neural tube. And we retain this neural tube. Some of it becomes very differentiated into different parts of our brain, and we retain an opening of the neural tube in us as adults that will be our ventricle system of our brain and surrounding within our brain. And so you have two lateral ventricles, a third ventricle, and a fourth ventricle. And so we retain this in our adult form and structure. And then we have something known as the growth cone. This is the growing tip of an axon. It's going to take up different types of uh, chemicals, um, and then use that as it continues to grow and develop. Um, but that is where we're going to stop for our nervous system lecture. As always, if you have any questions at all, please feel free to email me or reach out to me, and I'll be happy to help. And I will see you in the next video.